there's been a long-standing interest in potentially modifiable risk factors in the context of multiple sclerosis. We know that among the risks of developing MS, of course, there are genetic factors that appear to be different in different individuals and contribute to some of the risk, but there are also environmental factors, and amongst them, adiposity has been identified, or obesity has been identified as a risk factor for developing MS. And in this context, we were interested in what the biological mechanisms involved might be. And we've learned with the field over the years that there are a variety of molecules referred to as adipokines that are released in part by cells of fatty tissue, adipose cells. And these adipokines, which serve important roles in metabolism, of course, and relate to a variety of biological processes, also turn out to have very interesting immunological properties. And some of them have been identified as potentially pro-inflammatory, others as potentially anti-inflammatory. One in particular, adiponectin, has been identified at times as pro-inflammatory and other times as anti-inflammatory. And that has made it particularly interesting to try to understand. When we applied in the pediatric MS context, which of course is early development of MS, a unique window of opportunity to try to understand risk factors that are present around the time of the individuals actually acquiring multiple sclerosis. The observation when we screen serum of children with MS, as well as children with other CNS inflammatory conditions that are not MS, as an important other CNS inflammatory control and healthy controls, we identified amongst adipokines in the circulation that adiponectin was present in the children with MS at a higher concentration than the two other control cohorts. And we then, through a series of studies looking at the functional implications of that, were able to show that the adiponectin elevated in the serum of MS children can result in pro-inflammatory changes in myeloid cells, macrophage from the peripheral blood, which in turn could drive pro-inflammatory responses of T cells, including the T cell subsets that we think are implicated as pro-inflammatory and causing disease activity in multiple sclerosis. So the observation that adiponectin, at least in the pediatric MS context, appears to have pro-inflammatory, not anti-inflammatory properties, and that it mediates the pro-inflammatory properties through modulating myeloid cells and in turn inducing pro-inflammatory responses of T cells, could be part of the explanation for the implication of adiposity, or at least for us to think about how potentially modifiable risk factors in the context of MS development may be targeted in a beneficial way.